in this video lecture, we are going to discuss about the carbohydrates. Carbohydrates, these are simply the hydrates of carbon with the formula of CnH2ON. If you put the value of N equals to 6, then it will become C6H12O6. It is glucose, which is the most important sugar in biological systems. The carbohydrates, these are polyhydroxyaldehyde or ketones. If a molecule is having a single OH, it is monohydroxy. If it is having two OH, it is dihydroxy. And if it is having more than two hydroxyl, then it is called as polyhydroxy. So the carbohydrate, they are polyhydroxyaldehyde or ketones. Now, the simplest carbohydrate, the simplest aldehydic carbohydrate is glyceraldehyde, while the simplest ketone carbohydrate, it is the dihydroxyacetone. The types of carbohydrates, there are three major types of carbohydrates, that is monosaccharides, which do not get further hydrolyzed, oligosaccharide that gives 2 to 20 residues upon their hydrolysis. Third one is the polysaccharide that will give more than 20 residues upon its hydrolysis. Glucose, it is the blood sugar. It is the only source of energy for developing embryo in case of plant. In plant, during the embryo development, there is a layer which is called as alleuron. It releases an enzyme which is known as amylase. There is endosperm structure that provides nourishment to the developing embryo. The endosperm stores energy in the form of starch. Now this amylase, it will act on this starch, will carry out the hydrolysis of this starch and forms glucose. Then this glucose will mobilize towards the embryo cell. Next, the brain also obtains 80% of its energy from the glucose. Stereochemistry, D and L forms. D and L forms, these are on the basis of position of functional group on the penultimate carbon. Penultimate carbon is the second last carbon in the chain. It is the last carbon or ultimate carbon, and it is the penultimate carbon. If the position of functional group is like that, it is known as D form of glyceraldehyde. If the position of functional group is like that, it is known as L form of glyceraldehyde. Naturally occurring all sugars are D sugars, while naturally occurring all amino acids are L amino acids. Enantiomers, the isomers which are mirror images of each other, they are called as enantiomers. We can take the example of 4-carbon sugar, that is erythrulose. You can see over here, these two structures, they are the mirror images of each other and known as enantiomers. Next is the epimers. This is the structure of glucose having 6 carbons. 2, it is a hexose sugar, 4, 5 and 6. Now, the penultimate carbon is the carbon number 5, while the ultimate carbon is carbon number 6. Similarly, this is the structure of mannose, and the difference between these two structures is on the position number 2. Similarly, this is the structure of galactose, and the difference between these two structures It is on the carbon number 4. So, what are apimers? If there is a change in the position of functional group beside the penultimate carbon, then it is known as apimer. Here, the mannose is the C2 apimer of glucose, while the galactose is the C4 apimer of glucose. While these mannose and galactose, they are known as distereoisomers. What are distereoisomers? If there is a change in the position of functional group at more than one position beside the penultimate carbon. यहाँ अपन देख सकते हैं कार्बन नंबर 2 पे भी फंक्शनल ग्रुप की पोजीशन डिफरेंट है और कार्बन नंबर 4 पे भी फंक्शनल ग्रुप की पोजीशन में डिफरेंस है, तो इनको अपन कहेंगे डाइस्टीरियोआइसोमर्स और केवल एक ही जगह पर चेंज है, देन इट इज नोन एज एपिमर बिसाइड द पेनल्टीमेट कार्बन. So this is the concept of apimers and distereoisomers. Next is the hemiacetal and ketal. The sugar with 5 carbon or more, they readily adopt a ring structure. This is the structure of glucose. It is a 6 carbon aldehyde sugar, while this is the structure of a 6 carbon ketone sugar, which is known as fructose. The 6 carbon aldehydic sugar adopt a ring structure that is called as hemiacetal, that is a heterocyclic structure which contains 5 carbon and 1 oxygen. These are also known as the pyrene ring. While the 6 carbon ketone sugar, they adopt a ring structure which is known as ketales that contains 4 carbon and a oxygen, known as furane rings. Now, why these structure or why these we can say chain structure will convert into the cyclic structure or ring structure? The reason behind this is the angle between the carbon and carbon in the carbohydrate, which is not 180. It is 109. Due to association with such an angle, the hydroxyl group of penultimate carbon, they comes in front of the aldehydic or to the ketonic groups. This will act as a electrophile, will attack this carbon. There is a formation of new bond between the oxygen and carbon, and this double bond will convert into a single bond, and there is a presence of hydrogen over here. देख सकते हैं कैसे बनता है रिंग स्ट्रक्चर. ये ऑक्सीजन अपन बात कर रहे हैं जो यहां पे कार्बन नंबर 1, 2, 3, 4 and 5. So hemiacetal, it is 5 carbon and 1 oxygen ring. कार्बन नंबर 1 पे अपन देख सकते हैं ये कार्बन नंबर 1 है. यहाँ पे क्या है, यहाँ पे एक H है और एक OH ग्रुप है. So there is a H and OH group. Carbon number 2, there is H and OH. On carbon number 3, there is a reverse orientation. On carbon number 4, again there is H and OH. On fifth number carbon, there is a presence of this structure as well as this hydrogen. So there is this is the ring structure of glucose, or we can say it is a pyrene ring. Similarly, if we talk about the fructose, there is 4 carbon and an oxygen in the ring. Again, this is the oxygen. Similar intramolecular reactions takes place. The 1 number carbon, it will not participate in the ring. There is carbon number 2, 3, 4 and 5. On carbon number 2, there is a CH2OH group. And there is a presence of this OH. On carbon number 3, there is H and OH. Carbon number 4 also contains H and OH. On carbon number 5, that is the penultimate carbon, it is having a H and again the CH2OH group. This is the structure of fructose, ring structure of fructose, we can say. Now again there is a concept of anomers. There is a concept of anomers. And what are anomers? If there is a change in the position of functional group, if there is a change in the position of functional group on reactive carbon or carbon number 1, then it is called as the anomers. So anomer of this structure will be like this. This is carbon number 1, 2, 3, 4 and 5. First number carbon, there is a change in the position of functional group. This is called as alpha D-glucose. This is known as the beta D-glucose. And these alpha D and the beta D-glucose, they are called as the anomers. There is a change in the position of functional group on the anomeric carbon or the reactive carbon, that is the carbon number 1. Next is the concept of म्यूटा-रोटेशन. सिंपली पहले वाला जो कॉन्सेप्ट अभी अपन ने क्लियर किया उसी पे बेस्ड है, दैट इज द रोटेशन दैट अकर्स अराउंड anomeric carbon. The rotation around the anomeric carbon is known as the म्यूटा-रोटेशन. If we dissolve a sugar, any sugar, in the water will contain equal amount of alpha and beta form. So to reach the equilibrium, there is a rotation around the anomeric carbon, that is known as the म्यूटा-रोटेशन. किसी भी शुगर को आप सॉल्यूशन में डालते हैं तो सॉल्यूशन में अल्फा और बीटा फॉर्म की इक्वल अमाउंट रहेगी. उस इक्वल अमाउंट तक पहुंचने के लिए जो रोटेशन होता है दैट इज कॉल्ड एज द म्यूटा रोटेशन. नेक्स्ट इज द oligosaccharides that will give 2 to 20 residues upon their hydrolysis, and the simplest oligosaccharide is the disaccharide that is made up of two monomers. We will start with the sugar that is called as the maltose. This maltose, it is the disaccharide of alpha D glucose. It is the ring structure of alpha D glucose. It is the ring structure of alpha D glucose. The two monomers of alpha D glucose, they will come together. There is a intramolecular reaction. There is a removal of, or we can say there is a condensation. There is a removal of water molecule from here, and there is formation of new bond between two monomers, which is known as maltose. Now this is the carbon number 1, 2, 3, 4, 5 and 6. Here carbon number 1, 2, 3, 4, 5 and 6. Second type of sugar, it is the milk sugar, which is called as lactose, and the lactose is disaccharide of beta D galactose and alpha D glucose. It is the structure of beta D galactose. Galactose is the C4 aptamer. There is a change in the position of functional group on the कार्बन नंबर 4. देख सकते हैं अपन स्ट्रक्चर इसका और इसका, यहाँ पे आपको 4th नंबर पोजीशन पे चेंज देखने को मिलेगा. इट इज द स्ट्रक्चर ऑफ बीटा डी ग्लूकोज, क्योंकि हाइड्रोक्सिल ग्रुप ऊपर की तरफ है इसलिए बीटा डी ग्लूकोज गैलेक्टोज है. नेक्स्ट स्ट्रक्चर इट इज ऑफ अल्फा डी ग्लूकोज. देन अगेन हियर इज ए intramolecular reaction that is known as the condensation. There is a removal of water molecule and formation of a new bond between the two sugars. This is known as lactose. Next type of sugar, it is the sucrose, and the sucrose is the disaccharide of alpha D glucose and beta D fructose. It is the fructose structure and it is the structure of glucose. Now the glucose, there is carbon number 1, 2, 3, 4, 5 and 6. Here the carbon number 1, 2, 3, 4, 5 and 6. Again, similarly, there is a condensation reaction. The water molecule, it comes out and there is a formation of bond between the two sugars. These bonds here, in the maltose, this bond is called as alpha 1, 4 O glycosidic bond. It is a O type of ग्लाइकोसिडिक बॉन्ड. बॉन्ड्स जिसके बारे में अपन ने कल बात की थी. यहां अगर बात करें तो अपन कहते हैं देयर इज ए बॉन्ड दैट इज नोन एज बीटा 1, 4 O glycosidic bond. O glycosidic bond means it is formed between the sugar and sugar molecule. Next, here it is the bond that is known as alpha 1, 2 O glycosidic bond. It is alpha 1, 2 O glycosidic bond. Now there is a concept of reducing and non-reducing sugar. The sugars which have free anomeric carbon can reduce the solution of copper and ferrous. They can reduce the solution of copper and ferrous, and they are called as the reducing sugar. Means that sugar that are having free, we can say free anomeric carbon. और रिएक्टिव कार्बन। तो अपन देख सकते हैं डाइसैकेराइड्स के अंदर फ्री एनोमेरिक कार्बन यहां पे प्रेजेंट नहीं है। माल्टोज के केस में देखें तो बिल्कुल फ्री एनोमेरिक कार्बन अवेलेबल है। यहां देखें तो बिल्कुल फ्री कार्बन एनोमेरिक कार्बन अवेलेबल है। सो वी कैन से दैट द माल्टोज एंड द लैक्टोज दे आर द maltose and lactose, they are the reducing sugar, while in case of sucrose both the anomeric carbon, that is the carbon number 1 of glucose and the carbon number 2 of fructose, they are involved in the bonding. That's why they are known as the non-reducing sugar. So it is the concept of reducing and non-reducing sugar. Next are the derived sugar. These are the sugars derivative. They are two types, amino sugars and the acidic sugars. अगर acidic sugars की अपन बात करें तो ये स्ट्रक्चर आप जो देख रहे हैं ये ग्लूकोज का स्ट्रक्चर है. If this CH2OH it is replaced by the acidic group, then it is known as the glucouronic acid. And if there is a replacement of this H with the acidic group, then it is called as the adiuronic acid. It is very much simple. So there is a glucouronic acid and there is a adiuronic acid, which are acidic sugars. If we talk about the amino sugars, if there is a replacement of this OH with NHCO CH3, that is acetyl group, then it is called as the N-acetyl glucosamine. It is the structure of N-acetyl glucosamine. While if there is a replacement of this OH with this, then it is called as the N-acetylmuramic acid. The sugar which is having the acetyl group on the carbon number 2 and there is a position of this acidic group on the carbon number 3, this is called as the N-acetylmuramic acid or NAM, which plays an important role in the bacterial cell wall formation. Similarly, if there is only presence of this acetyl group over here, it is known as N-acetyl glucosamine. This sugar also plays an important role in the formation of peptidoglycan cell wall in case of bacteria. So these are the derived sugars. Next is the polysaccharides. Polysaccharides are those carbohydrate which gives more than 20 residues upon their hydrolysis. They are mainly of two types, homopolysaccharides that give single type of residues upon its hydrolysis, and if the polysaccharide gives more than single type of residue upon its hydrolysis, it is known as heteropolysaccharides. Homopolysaccharides, they can be further divided into two types, that is the storage homopolysaccharide and structural homopolysaccharide. बेसिकली जो स्टोरेज होमोपॉलीसैकेराइड्स हैं ये एनर्जी प्रोवाइड करते हैं, वाइल द स्ट्रक्चर जैसा नाम से ही लग रहा है ये स्ट्रक्चर्स बनाने में काम आते हैं. पहले बात करते हैं अपन स्टोरेज होमोपॉलीसैकेराइड्स की. फर्स्ट एग्जांपल लेते हैं, दैट इज ऑफ द स्टार्च. स्टार्च बेसिकली इट इज द पॉलीमर ऑफ अल्फा डी ग्लूकोज. Starch is the polymer of alpha D glucose. It is of two type, that is amylose and amylopectin. Amylose की अगर बात करें तो यहाँ पे केवल अल्फा 1,4 लिंकेजेस होते हैं अल्फा डी ग्लूकोज के बीच में, while in case of amylopectin, beside this alpha 1,4 there is another type of linkage which are known as alpha 1,6. यहाँ स्ट्रक्चर में भी अपन देख सकते हैं, these are the monomeric units of alpha D glucose and there is a bond which is known as alpha 1 and 4, जिसके बारे में अपन पहले भी बात कर चुके हैं. These are alpha 1,4 glycosidic linkages. This is the structure of amylose. While in case of amylopectin, after 15 to 20 residues, there is a formation of alpha 1,6 linkages. Branch structure बनेगा. कैसे बात करते हैं? It is the 6 number carbon. It is the 1 number carbon of glucose. There is a formation of glycosidic bond. We can say the condensation reaction takes place. H2O comes out, and there is a formation of a alpha 1,6 linkage between these two sugar, which carry out the branching in this starch. So we can say that the amylopectin, it is a branch structure, while the amylose, it is a linear structure. What is the advantage of having these alpha 1,6 linkages? Like starch deposits in the form of concentric rings due to the presence of these alpha 1,6 linkages that occupies lesser spaces, as well as the starch, it is identified by the iodine due to the presence of these grooves, because the size of iodine is identical to these grooves that can be easily identified by the iodine. So this is the reason why the starch can be identified by the iodine due to the presence of these alpha 1,6 linkages. Now, next type of we can say sugar storage homopolysaccharide, it is the glycogen, that is also known as the animal starch. The structure of glycogen is similar to the structure of amylopectin, having both alpha 1,4 and alpha 1,6 linkages, but it is more branched as compared to the amylopectin. Next type of storage homopolysaccharide is inulin. It is the polymer of alpha D fructose. It is a polymer of alpha D fructose having alpha 1,4 linkages. Inulin, it is water soluble and it is used to determine the glomerulus filtration rate. So it is an important sugar. Next is the structural homopolysaccharides, and we are taking two examples over here, that is of cellulose and chitin. Cellulose, again, it is the polymer of alpha D glucose. It basically forms the cell wall of plants. There is no known animal beside, or we can say except, the silverfish and termites that can digest this cellulose. So there is no presence of cellulase in any organism beside these silverfishes and the termites. While in case of ruminants, होता क्या है कि the ruminants, they have symbiotic bacteria, we can say, in their gut. These bacteria, they have ability to release the enzyme cellulase. The enzyme cellulase, it will act on this cellulose. As we all know that it is the polymer of alpha D glucose. So it will carry out the breakdown of this cellulose into the glucose. Then this glucose is utilized by the bacteria for their metabolism, and as a byproduct, these bacteria, they releases the fatty acids. And these fatty acids, these are of no use for the bacteria, but they can be utilized by the ruminant for the energy generation. So this is all about the cellulose. Next is the chitin. Chitin, it is the we can say it forms the cell wall of fungi. It is, again, we can say, the polymer of N-acetyl glucosamine. The bacterial cell wall, it is made up of peptidoglycan, which is the polymer of N-acetyl glucosamine and N-acetyl muramic acid, while the fungal cell wall is made up of chitin. Chitin is the polymer of an acetyl glucosamine having beta 1,4 linkages. Next is the heteropolysaccharides. The heteropolysaccharides, they are mainly divided into four types. That is the hyaluronic acid, heparin, chondroitin sulfate and keratin sulfate. पहले बात करते हैं अपन हाइलयूरोनिक एसिड की. सो यहाँ पे ये हेटेरोपॉलीसैकेराइड है. क्यों? क्योंकि इसमें एक एसिडिक शुगर रहेगी और साथ में एक रहेगी अमाइनो शुगर्स, जिनके बारे में अपन बात कर चुके हैं पहले भी, एसिडिक शुगर्स एंड अमाइनो शुगर्स के बारे में. तो यहाँ हाइलूरोनिक एसिड में अमाइनो शुगर रहेगी एन एसिटाइल ग्लूकोसामीन और एसिडिक शुगर यहां पे रहेगी ग्लूकोयूरोनिक एसिड. Glucouronic acid. While in case of heparin, again the amine sugar, it is the N-acetyl glucosamine, but there is a presence of id-uronic acid in place of glucouronic acid. Next is the chondroitin sulfate. It is the, we can say, the polymer heteropolysaccharide of N-acetyl galactosamine. It is having the N-acetyl galactosamine. Galactose is the C4 epimer of glucose. So there is a N-acetyl galactosamine plus there is a glucouronic acid. Now, in last, that is the keratin sulfate, there is a absence of acidic sugar. Simply there is a galactose and there is a presence of N-acetyl glucosamine. So these are the heteropolysaccharide. Thank you.